I am a student from the University of Toronto in Dr. Hannes Rost's lab, where we focus a lot on personalized medicine and a lot more on proteomics and mass spectrometry methods development. So to begin with, I want to first start out with why we're interested in utilizing the plasma and looking at the plasma. The plasma is a liquid component of the blood, and it offers several advantages as a sample for measuring analytes compared to other sources such as tissue or urine. Namely, it's a systemic representation that the plasma circulates throughout the entire body, acting as a conduit that delivers all of these molecules to various tissues and organs. And as a result, it carries valuable information about the overall physiological states, including any sort of disease-related changes. And so if we analyze the plasma, we can obtain some of this systemic representation of the actual molecular landscape that might provide us with some insights into the body's responses to diseases like cancer. And then for 2 and 3, they're kind of similar in that the accessibility and convenience. It's fairly easy to acquire some blood samples, especially the plasma part of the blood. It can be easily collected in any sort of clinical setting if you just go to a clinical laboratory to get your blood taken. And it's a fairly standardized procedure of how you collect blood and how you process it. And then for the third point, it's non-invasive sampling. So it's not, we don't have to take the tissue from the patient. You can just, again, collect from the blood. So you don't have to do any sort of surgery or cutting, which is valuable to have these sort of biopsies, but it's also not easy to get these in a term of a longitudinal fashion, which brings me to the next point of dynamic monitoring, where we can actually collect these plasma samples to identify and reflect real-time changes within the body, which makes it really suitable for this longitudinal monitoring. So we can serially sample plasma from patients and track the dynamics of how these analytes are changing over time. And then the third point is actually, the next point is a lot of these analytes are actually coming from different organs in the body. So it's representing, organ-specific changes. And that's like how the proteomic landscape actually measures analytes different, originating from different parts of the body. And as the blood is circulating throughout the entire system, it comes into contact with various tissues and organs, which carries diverse range of molecules derived from different sources. And this can be utilized for information to identify specific changes from different organs and contexts that it comes into play. And lastly, because of all of this, basically, it just really allows for really a great set of biomarker discovery, because it basically facilitates the identification of these biomarkers, which are measurable indicators of any sort of changes in the biological processes or disease states. And if we analyze these expression profiles of the proteome and plasma, we can then potentially identify which specific analytes are associated with a specific disease, including cancer. And then we can utilize these biomarkers for early diagnosis, prognosis, or even predicting any sort of relapse or recurrence of the disease. And with that all being said, hopefully we can see why we want to measure the proteome from the plasma and why it's important, because it contains this wealth of information that we can use to understand biology. It acts as a snapshot of the current state of biology for a specific individual, and it reflects the actual dynamic changes that occurs in different organs. And this diverse range of analytes derived from the different organs that circulate through the bloodstream provides basically a holistic view of the actual physiological state and pathological state. And by analyzing these proteomes, we can uncover valuable insight into disease and the overall health status. But as I mentioned earlier, from other talks, generally, these biomarker discovery approaches is based on a broad-based approach where we collect a bunch of different patients and a bunch of different samples from a large cohort of patients, which we then analyze and measure those proteins in the body to try to identify what's associated with a specific disease or condition. And this is generally done through the analysis of this wide population to try increase the likelihood of identifying some set of comprehensive biomarkers that can provide valuable insights to the disease mechanism of the disease. And it's generally using either some sort of model to identify these differential express proteins and then trying to best separate the patients into different clusters of subgroups. However, this traditional one-size-fits-all approach to patient care, which includes surgery or chemotherapy or specific drug treatments, has shown to be effective. There are still several challenges that remain, specifically recurrence, which is often linked to some sort of drug resistance, which enables the tumor to metastasize and contribute to cancer progression. And so just in this example cartoon here, you have one patient who is diagnosed and then they get treated with drug A, and for them it works, so their life expectancy is improved. But for these other two patients in red and purple, they've given the exact same drug, but maybe it works for one patient, but then they get some sort of recurrence and then they end up passing away. And it's the same case with the purple case, where just the drug doesn't work for these specific two patients because it's not tailored for their specific biology. And this is kind of where we strongly believe in the power of the N-of-1 personalized medicine approach, where we can capture an individual's proteome profile at diagnosis and conduct subsequent plasma proteomic measurements where we can detect the changes in specific pathways over time. And this enables us to identify these tailored treatments that may yield better outcomes for each patient, ultimately enhancing the quality of life. And then furthermore, because we can continuously monitor and acquire blood samples even after treatment, we can assess the patient's response to the treatments and potentially use this longitudinal information to try predict and prevent actual cancer relapse or recurrence. I want to shift focus to the actual pilot plasma proteomic study involving Shirley and Vanessa. So in this subset of the data, we have 6 random donors that are healthy individuals. We have 1 sample from Shirley, and then we have 4 different longitudinal time points from Vanessa. So before I delve into some of these figure slides of the actual data, which we've already seen, I guess, some of these explanations of how we go from data to analysis. I just wanted to give you like a high level overview of how we go from identifying a cluster of patients based on their proteomic profiles. And so we can imagine like this dense forest as a collection of proteomes identified for each patient. And this abundance of information with these proteomes actually presents a challenge in separating patients on specific diseases or condition, just because of like the amounts of biological variation in each patient. There may be some overlapping subproteomes and some noise and technical variation. So generally, what people do is they employ some sort of feature filtering techniques to kind of navigate through this dense forest of proteomic profiles and to identify the most informative and relevant features that contribute to significantly distinguishable information that can discriminate these different patients. And by selecting these key features, we can create these clusters that reveal how patients with similar proteomic profiles cluster together, which can provide us insight into the actual disease of subtypes or response groups. And then this approach helps us unveil meaningful patterns and relationships within the data, which may enable more precise understanding of patient groups and more personalized treatment strategies. And so in this example, we've already had some insight into the use of PCA. So here I'm just showing basically a combination of a random forest feature selection to reduce the proteome space, and then applying a principal component analysis to visualize the clustering of the patient proteomes. And basically, we've already heard this before, but the PCA basically just aims to compute some lower dimensional representation of the data that finds some orthogonal dimensions that captures the most amount of variation, which is basically just the dimensions that contain the most amount of information. And so the X and the Y axis here represents the first and second principal components, respectively, with their associated percentages of variance explained by the data. And the blue points correspond to healthy patients, while the red points represent the two cancer patients, Shirley, which is labeled as P21, and Vanessa, which is labeled as P25. And so we observe that the unrelated healthy donors tend to cluster together. So they clearly have a distinct proteome profile compared to the cancer patients. And notably, the middle time points of Vanessa, which is P2 and P3, they cluster together, which indicates the power of this longitudinal profiling and consistency of a patient's own proteomic profile that is still similar to themselves over time. However, it is still interesting to note that the latest time points of Vanessa, which is time point 4, clusters relatively close to Shirley at the very top quadrant there. And so we're currently delving further into why this might be the case and why this specific sample time point is very close in proximity to Shirley's time point, like how their proteome profiles are actually correlating or closely matching together. This analysis basically just demonstrates how feature reduction and PCA can reveal meaningful patterns in patient proteomic data, which can aid in the identification of distinct clusters and provide insights into disease progression and patient similarities. So in addition to identifying clusters, we are also able to demonstrate the ability of just identifying differentially expressed proteins using simple linear regression models. And we can clearly see that there are patient-specific profiles for key differentially expressed proteins in the heat map. However, for the purpose of the presentation, I won't really delve into the specific differentially expressed proteins in this data set. Instead, I would like to highlight the power of the N-of-1 analysis, which focuses on a single individual Vanessa over time. And this analysis will allow us to closely examine Vanessa's proteomic profile and its dynamics, providing more valuable insight into her own unique responses to disease progression. And by monitoring her changing proteome profile, we can actually gain a deeper understanding into the changes occurring at the molecular level and potentially identify specific pathways of proteins that are associated with progression of her disease state. So to analyze the longitudinal proteomic profile of Vanessa and in order to identify any sort of trends, I conducted just a simple Pearson correlation analysis. And so the x-axis here is just representing the actual Pearson correlation coefficient, which is indicating the strength of the correlation between a protein's intensity over time. And the y-axis is just displays the negative log 10 p-value of the actual correlation test. And this correlation analysis is basically just looking for positive or negative trends of proteins that are changing over time for Vanessa's proteome profile across those different 4 time points. And if we just apply a 5% p-value cutoff and a Pearson correlation cutoff of 0.8, we observe numerous proteins that exhibit either positive correlations, and then there's a few that show a smaller subset of these proteins that are negatively indicating an inverse relationship over time. And so an intriguing finding from our analysis that we do see from these 279 proteins is that we do identify this potential pathway associated with the cytokine storm, which was mentioned in an earlier talk. And this pathway just really encompasses, a series of interactions and signal events from cytokines, which are these small proteins involved in immune response inflammation regulation. And so the presence of this cytokine storm may suggest some aberrant immune response in Vanessa's longitudinal proteomics data. And the cytokine storm refers to basically just the excessive and uncontrolled release of these cytokines, which is often associated with severely inflammatory conditions. So this is interesting because it raises questions about the underlying mechanisms and potential implications of Vanessa's health during the time period between these 4 different sample measurements. And so if we try to understand how this pathway is being activated, it could provide us with some valuable insight into the actual immune dysregulation or inflammatory processes that occurred in her condition. So in addition to actually identifying an overall cytokine storm pathway, we can actually further analyze these temporal profiles of these 270 hit proteins to try and cover subpathways or modules that exhibit similar longitudinal patterns. So we can cluster these proteins based on their actual expression profiles over time to try to identify groups of proteins that share very similar or common behavior and potentially participate in coordinated biological processes. So if we just utilize some sort of fuzzy clustering, we can observe that the proteins clustered into 4 different distinct temporal profiles. The first 2 clusters were a more immune system-based response related to either signaling by interleukins or some immunoregulatory interaction between the lymphoid and non-lymphoid cells, respectively. Whereas clusters 3 and 4 showed some sort of enrichment in extracellular matrix organization and RNA metabolism. And so this clustering analysis really just allows us to explore functional relationships and dynamics among these identified proteins. And by grouping them together into actual distinct modules or subpathways, we can gain a bit more granular understanding of their interactions and roles within the broader cytokine storm pathway. So this modular approach allows us to capture these finer grain details in uncovering potential regulatory mechanisms or functional subnetworks within the larger pathway. And so basically, just by combining the identification of the overarching pathway of interest with these clustering temporal profiles, we can uncover the complexity and heterogeneity of Vanessa's proteomic landscape. And this integrated approach enhances our ability to decipher and intricate interplay of proteins and pathways involved in her longitudinal profile. So in conclusion, I hope to have shown you that plasma proteomics in personalized cancer care provides valuable insights into disease-related changes and longitudinal monitoring. And analyzing the proteome of the plasma captures these organ-specific changes, which may facilitate biomarker discovery and enables personalized treatments. And furthermore, this N-of-1 analysis approach allows for the identification of these longitudinal temporal profiles where we can identify pathways or proteins of interest for a specific individual's condition, which may reveal dynamic changes in the proteomic landscape. But moving forward, we currently have an ongoing prospective cohort study where we actually have included 3 additional cancer patients that are diagnosed with either pancreatic cancer, glioblastoma, and myeloma. And so this is just a preview of the initial rounds of analysis of this data set, where we just present a hierarchical clustering dendrogram of their proteomic profiles. And interestingly, like we really do see some distinct proteomic profiles that group the cancer patients specifically together, while the healthy controls and repeated measurements of some of these healthy controls actually also cluster to themselves. And so this is currently ongoing, and we're still delving a bit more into this. So first, I would like to thank you all for listening. I'd like to thank everyone in RTPP for allowing me to also present. And this is, I think, really cool and interesting work